Hello and welcome to this molecular science presentation. I'm Dr. Noreen Bhatti, and today I'll be walking you through the journey of the coronavirus in its successful pursuits in its infiltrating mechanisms of unsuspecting host cells in the human body. The viral mechanism of action and the manner in which coronavirus evolves have very important implications for therapeutic strategies, and this understanding would be key to keeping up with emerging COVID-19 variants, particularly when researching therapeutic interventions. Therefore, with a molecular and pharmacological lens, I have decided to talk through the molecular basis of COVID-19 and its evolution. So let's get right into it and start with this visual that I've decided to use for my introductory slide. So the image you see here is a simplified 3D representation of a virus particle, or we can refer to this as a virion. Now, this spherical structure is specially designed and adapted with multiple features to aid its function during viral attack. An integral component is the spike glycoprotein, which we can see here as these protrusions that are studied across the periphery of this spherical structure. And these are coloured in a pale reddish colour or a peach colour here. The component which makes up the blue bilayer that we can see here is the viral envelope, and this consists of both lipids and proteins. Now, this structure is pivotal for housing and compartmentalizing the genetic material, which we can see as this yellow ladder-like structure protruding externally and escaping its lipid coat. This is the representation of the genetic material, or the viral RNA. So the genetic material will hold the instruction or the code for the virus to form multiple copies of itself during viral replication. So you may be wondering why the code is being expelled from the coat in this particular image, and the reason for this will become apparent further into this session. So the first part of my talk will address viruses as a whole, then I'll present descriptions of the key structural features of COVID-19 in further detail. This will aid in the molecular journey of how COVID-19 infects its host, so essentially the infiltration mechanism and hijack of host cells, which is responsible for bodily infection. I'll then discuss the genetic evolution of the virus, aided by an example of a known mutation. We continually see emerging variants, so this understanding on a molecular scale may prove useful. In line with this understanding, I will discuss how variants arise due to mutations and also their implications on the effectiveness of vaccines and therapies. So before I talk about COVID-19 and its mechanism of action, I think it may be useful to talk a little bit about viruses as a whole. So viruses are considered the most abundant biological entity, and the way I like to put it is that viruses are infectious agents that are capable of exploiting its host for its own survival. Now, they can attack a range of both prokaryotic and eukaryotic organisms, so they can infect cells such as bacteria, human and plant cells. And common examples that the audience will be familiar with are the common cold, influenza, Ebola, etc. So at the bottom of this slide, I have inserted some viral electron micrographs. Now, as a bit of background information, to obtain these images, an electron microscope is used, and that's a microscope that uses a beam of accelerated electrons as a source of illumination, so you can get images of very, very small structures. Now, as you can see to the left is an electron micrograph of a cluster of influenza viruses, each about 100 nanometers long. To put things into perspective, this is billionths of a meter long. As you can see, both the membrane and the lipid coat are visible. And if you follow me to the central image here, we can see a very different type of virus. And this is TMD, or tobacco mosaic disease. And this is, as you can see, quite a characteristic difference between the first image and the central image here. Now, to the very right-hand side is an electron micrograph of SARS-CoV. And if you look very closely at its structure, you can see these club-like protrusions at the periphery, and that's very similar to what I had described in my previous slide. These are the characteristic features of the COVID-19 virus, and it is what my talk will be focused on. The virus that causes COVID-19 is a type of coronavirus, which is a large family of viruses. Viruses and the diseases that they cause, they often have different names. For example, HIV is the virus that causes AIDS. Now, people often know the name of a disease, but not the name of the virus that causes it. So the familiar term coronavirus or coronavirus disease or COVID-19 that we can't seem to get away from is the name of the disease, and the name of the virus, which is actually the causative agent of that particular disease. In this case, it is severe acute respiratory syndrome coronavirus 2. And it's a mouthful, so you can see why it's been abbreviated to SARS-CoV-2. Now, this name was chosen because the virus is genetically related to the coronavirus, which is responsible for the SARS outbreak of 2003. However, while they are related, the two viruses are very different. Just a bit of background, viruses are named based on their genetic structure, and that is to facilitate the development of diagnostic tests, vaccines, and, for example, medicines. And diseases, however, they are named to enable discussion on, for instance, disease prevention, spread, transmissibility, or severity and treatment. Now, there are various possible modes of transmission for SARS-CoV-2, including contact, droplet, airborne, as you can see in the image to the right-hand side, which is just a representation of a virus within a droplet traveling within the air. This is known as airborne transmission. So there are various symptoms when this virus is acquired, and this includes coughs and fever, lower respiratory tract infection, even multiple organ failure, and it can range all the way through to not presenting with any symptoms at all. So the common clinical features are very varied, which poses a problem as this makes coronavirus diseases quite indistinguishable from other respiratory infections. So SARS-CoV-2 has a very characteristic structure, as I mentioned briefly earlier, and I've now displayed this as a complete cross-section of a virion on this slide to discuss the two main features of its architecture, which are pivotal for supporting its function to infect its host. So first off, a very prominent feature are the spike glycoproteins, which are embedded within the lipid bilayer that I spoke about earlier. And this protein protrusion is a familiar spike that studs the surface of the coronavirus, giving it the appearance of a crown to electron microscopy, which was displayed earlier. So the protrusions at the periphery that I had mentioned. Hence the term corona, which is Latin for crown. The next important feature of the architecture of the virus that I'd like to highlight is the enclosed genomic RNA, and that's encapsulated by the membrane. RNA essentially is short for ribonucleic acid, but it's just the instruction or the code for the virus to produce its proteins, allowing the virus to hijack human cells and turn them into virus factories, which I will describe in more detail in the subsequent slide. Essentially, RNA consists of approximately 30,000 nucleotides, and nucleotides are the building blocks of the genomic strand you can see here in yellow. And the nucleotides, when they're all bunched up together, will encode the structural proteins as well as non-structural proteins of the virus. And what do I mean by these proteins? Now here, some parts or components I have not described are shown. So the envelope, the M protein, E protein, and further along some more. Now these are important proteins to ensure that the virus is capable of surviving and replicating, and the code housed inside this lipid bilayer instruct the cell to create these proteins and hence allow the virus to survive. Now, these other components of the virion, which I have left out the detail of, are still very important, but the two I'd like to focus on today for this session are the spike glycoproteins and the genetic material, which are key features to understanding the mechanism of action for COVID-19 infection. So let's talk a little bit about the virus's mechanism of action. This is actually a very complex mechanism, but it's very interesting as well. So to orientate you, this is one virus particle, which is something that you are probably familiar with now. And here what we are looking at is the membrane of the host. So inside here is the cytoplasm of a host cell, and this could be a lung cell, such as a respiratory epithelial cell. Now, the important part of this is that the spike proteins embedded across the periphery are capable of attaching to a certain receptor that's already embedded within the lipid bilayer of a cell within the host. So this is a pre-existing receptor, and this is called the ACE2 receptor. ACE2 is an abbreviation for angiotensin converting enzyme 2, and it acts as the receptor for entry of the virus into the target cell, a bit like a lock and a key. So we can think of this as the cell entry receptor. And if I scale this up, we can see here the virus particle and its protrusion, which is the spike, is latching on to the ACE2 receptor, which is already part of the lung cell or the host cell. Now, this mechanism, when this first part happens, it instigates a cascade of events involving other proteases, allowing the fusion of the virus membrane to the host membrane. And we can see that happening in this diagram here. Further to this, this allows the viral RNA to be injected into the cytoplasm of the host. And we can see that the virus hasn't got any other machinery with it to replicate itself, but what it does is it hijacks the machinery pre-existing within the host cell in order to create the viral proteins. And again, a cascade of other events occur, such as the use of proteases and other enzymes to allow the virus proteins in order to be generated. When it regenerates itself, it forms into a novel virion, which we can see here. It's compartmentalized into a vesicle and then transferred to the membrane, where it is released from the cell or undergoes exocytosis. So we can see that this is a novel virion, which is a replica of what was originally started with. Now, what's important is that all of this stress that's occurring within the cell, it's not normal for this host cell. So this stress to the endoplasmic reticulum and other organelles can cause the cell to die. And this can be part of why symptoms are seen. So as a very quick summary, the virus will come along and attach to the surface protein, which is the ACE2 receptor, and this will then cause the virus to hijack the host cell machinery in order to survive and replicate. And in doing so, it will destroy the host. Now, although a complex process, if nothing else, I think the key idea to take away from this particular slide is that the S protein, or the spike protein, is the key for the entry of the virus. So very generally, the genome is the genetic blueprint of an organism which provides the instructions to its production. Here I provide a schematic representation of the genome organization of the virus. Essentially what this is the section of the viral backbone or genetic material which encodes the information for the production of various viral proteins. Now, if you remember from earlier, the single-stranded RNA enclosed within the virion was the code. And when this is read by the cell's machinery, it can create all types of proteins. Now, although a complex image, I didn't want to leave out the details of the viral genome to give the audience a view of the complexity of the virus. Now, each region you see here, as shown here in the coloured boxes, they code for the synthesis of the following 4 main structural proteins, including the spike, the envelope, the membrane, and the nucleocapsid. There are also non-structural genes called accessory proteins and other non-structural genes. Now, importantly, any change to the genetic code that I've shown here can lead to a mutation. So we've just talked about mutations. So what are they? A mutation is a change to the genetic material. It could either be DNA or RNA, and potentially that could alter the overall protein sequence. Now, the structure of the S protein here is shown, and this is, as I mentioned in the previous slide, this is the genetic material. But if I was to expand on this and look at the protein structure which is created or generated from this code, we can see this polypeptide sequence, again looking very complex, but the essential bit is the part of the region of that polypeptide chain which is going to fold up into a protein called the receptor binding domain. And the receptor binding domain is abbreviated to RBD, and RBM is the receptor binding motif here. Now, I'm just going to back up a bit. So the sequence that we can see here is a polypeptide sequence, meaning that it's going to fold up into a protein, and in this case it would be folding up into the spike protein, and this is the code which would generate that polypeptide sequence. The point of this slide is to say that it's very common to have a mutation within the RBD, and then this is an important part of the molecule of the spike protein, because as we saw earlier, the spike is really important for attaching to its host. So any kind of change within the RBD, or mutational change, may cause a functional change of the spike protein, hence cause a change to its function. The numbers here are showing the positions of any certain amino acid within that polypeptide region. And a single change which occurs within this protein structure can lead to a huge effect. So to illustrate the scale of the mutation, I have provided here a blueprint of the spike protein, which is essentially a part of the code for the S protein. And we saw earlier that S protein is studied on the surface of the viral envelope. And in order to generate this protein, the code would have to be read. Now, to put things into perspective, this is just a section of the code. The entire genome of the SARS-CoV-2 virus encodes around 10,000 amino acids, which is what each of these letters are referring to. And the sequence of letters, which is read by the cell, is what generates the viral proteins. Now, importantly, any change within any one amino acid could cause a mutation. So at a certain position, I know it's very small on this slide, but you can see any position that changes, it can cause a new mutation. And a single amino acid change can actually have a huge effect, a huge and significant effect on the viral protein and how it functions. So for example, in this case, it could cause a change in the aggressive character of coronavirus, and so be considered as a new strain or a new variant. So we've understood a little bit about mutations. As I mentioned, it's a change to the base sequence of the genetic material of DNA or RNA. I want to walk through an example now so that we can understand in a bit more detail how the mutations occur. Again, I'm displaying the genetic material shown here, and then the protein sequence of that genetic material when it's read. The receptor binding domain has been highlighted of one of the subunits, which is the S1 subunit. Now, if any kind of change occurs within this particular region, it could cause a change in the function of that particular protein. And one example, which is the E484K mutation, which you may or may not be familiar with, is a biologically relevant or important mutation as it occurs within an important structural protein, as we've spoken about, which is the spike protein. Here I've given the protein sequence, or the amino acid chain sequence of the normal or original protein, and then the mutated version, which has been highlighted in red, and that is a substitution mutation causing certain functional changes to the protein conformation, thus changing perhaps the function or even the aggressive character of coronavirus, and it can be then considered a different type of strain or a different variant.
So E484K is a substitution mutation arising from a genetic change, which occurs in the receptor binding domain of the S protein. So specifically occurs at position 484 in the polypeptide sequence, and that at that position the E is substituted with the K, which you can see here. So what is E and what is K? So essentially these are shorthand for amino acids, and any change in the amino acids can cause a change in the sequence, i.e. the polypeptide sequence, which essentially will create the final protein product. As you can see here, the amino acid E is glutamic acid, and that's been substituted with K, which is abbreviated for is the abbreviation for the lysine amino acid. So what is the implication of this E484K mutation? Now this substitution is going to cause different conformational changes within the final protein structure, which you can see a representative image of here once that polypeptide has been folded up and the relevant bonds have been made. So the overall spike protein and its function may be affected due to this substitution. The E484K mutation has been shown to be associated with escape from neutralizing antibodies produced by the body against SARS-CoV-2. So essentially, this means that this small mutation is capable of changing the protein structure of the spike, so much so that it can allow it to evade the function of antibodies, and this may also extend to therapies and vaccinations. But how is this possible? The next part of this session will address the conformational protein changes elicited to cause this functional change. So I'm now hoping we've come to a stage of this session where you have an idea of what a mutation is. So even if it's a very general idea. So if this question was posed to you, what is a mutation? What would your thinking be? I think what would be most beneficial is if, rather than forming a complete answer, have some keywords in your mind when forming an answer to this type of question. In subsequent slides, we can have a look to see if they are correct. So I now show a 3D representative protein sequence, and this protein sequence is different from the one displayed earlier because it's not a single chain. It's now been folded up into the conformation that it's needed to perform its function. In particular, this one is of the spike protein, and just to orientate you, the spike protein here is in the grey structure, the ACE2 receptor is in green, and within this structure you can see that there are coloured dotted regions, and those are the places of the mutations which may occur. And the one that is familiar for this certain session is E484K mutation. Of relevance, this mutation is occurring at the binding site of where the ACE2 receptor will latch onto this spike protein. So if you have a little bit of a think about if this changes or if this mutation occurs, then it may also affect the way that the ACE2 receptor will bind to the spike protein, which is essentially what happens with the E484K mutation, because it alters the shape of the protein conformation and it alters how it will interact with its normal target, which is the ACE2 receptor. That will affect the affinity, virulence, infectivity, or transmissibility. Could be any one of those. As well as that, it will also affect how it interacts with therapeutic targets, such as vaccines, antibodies, drugs, and decoys as well. I have put here two types of variants, so the South African variant and the UK variant, and this is to show that there are various types of mutations that can occur. So we can see that the common mutation is N501Y in both of the variants, but there are also other mutations which are different. So the E484K mutation is within the South African variant, and the UK variant has a 69 to 70 deletion mutation instead of a substitution. So this means that it will cause differences in the function and then the performance of this certain protein. In a number of persistent infections, notably HIV, mutations emerge at an extremely high rate because of the very high replication rate. For example, approximately 10 to the exponent 10 new variants which emerge per day. As we start to get standardized drug deployment, resistant mutations will likely arise. As a rule, evolutionary adaptation happens due to selection pressure, which is when an organism's environment changes to favor certain variants over others. Now, natural selection results in favorable genes being passed on to the next generation in proportions which will differ from those in the present generation. Now, to illustrate this point, have a look at the diagram to the right of this slide. Here, the mixture of diversely colored circles represents the variety of genes which various COVID-19 variants may contain. Now, these are most likely not detectable until a selection pressure arrives. The pressure in this instance could be a vaccine or a therapy. And at the point of exposure of the therapy, what will happen is that the variants with no mutation or no selective advantage will be killed off. However, those variants with a mutation, which will confer benefit to the virus, in this case not being destroyed by the therapy, they will survive and replicate. And that replication means that they will also pass their genetics with the mutation to the subsequent generation, shown here as G2. And as you can see here, the G2 generation have the exact same resistance mutation, which are the red circles here, and that allows them to survive any future exposure to that therapy. So successful therapies could cause the virus to evolve resistance, and resistance mutations will quite likely arise. The same will apply to the spread of natural immunity and vaccines. Over time, variants, as I mentioned, it may escape the vaccine's target or a therapy's target. For instance, the E484K mutation may escape from the host immune defense. So what's important to also note is that any kind of selection pressure, it doesn't force the virus to mutate, but if a mutation conferring resistance to a drug happens to arise in a virus, which is inside somebody who is being treated with that drug, then the mutant could proliferate and go on to infect another person. And then this could spread far and wide. This is known as escape. So we've been through the detailed mechanism of how COVID-19 will infect this host to cause the overall infection. What the next question is what stage would be an effective therapeutic target? So of the mechanism that has just been described, where would be the best way or the most effective way to target the virus to prevent viral replication? Now, as you may have already guessed, there are multiple approaches to preventing SARS-CoV-2 from replicating. But one of the main ways is to block the spike protein. And we can see this spike protein structure here in green, which is binding to its dimeric ACE2 receptor. If we were to block this interaction, it would prevent the virus from latching on and fusing and going into the host cell. I'm going to talk through a few strategies which have been deployed and are being researched to prevent this association. So firstly, engineered ACE decoys. So ACE2 mutants are engineered to bind to the spike protein more tightly than native ACE2. And what this does, it inhibits the actual SARS-CoV-2 infection from binding on to the host receptor and entering the cell. Next up are monoclonal antibodies. So spike protein specific antibodies will block the spike protein and prevent viral fusion. Monoclonal antibodies are essentially laboratory-made proteins that mimic the immune system's ability to fight off harmful pathogens such as viruses. Then we have small molecules or peptides, and these are molecules which are created to inhibit ACE2 by binding by S protein. Again, another way to block or inhibit that association. And then we also have vaccine-elicited antibodies. And these are naturally produced antibodies which are specific to the S protein, and again, the same mechanism to block ACE2 binding sites. So there are currently a few therapeutic approaches which are focused on blocking this particular association, which is the SARS-CoV-2 binding to its key receptor, ACE2. But it's important to also mention that there are also other approaches that occur perhaps within the cell. So once the virus has latched on, fused to the membrane, and then it goes into the cytoplasm, other components which are important for viral survival and replication, such as protease inhibitors, can be utilized to prevent the virus from surviving and replicating. So just coming back to the question that was posed earlier, I provided a very simple definition of what a mutation is. So a mutation is an alteration in the genetic sequence. And I've also provided a few keywords that I have also used in this presentation that can form part of the answer to this question. So as a quick summary, I'm hoping this session has allowed you to understand a little bit more about what mutations are. So any changes to the genetic backbone, either DNA or RNA, is called a mutation. And as we saw earlier, there are various types of mutations, such as substitution or deletion mutations, and the nature of each of these mutations are going to then determine the function of the final protein. And as you can imagine, the overall structure of that protein may be changed because of that particular mutation. The combination of a mutation and the selection pressure is what will generate new variants if they were to arise. And these variants, depending on the type of mutation that has been acquired, they may evade vaccine function, and that is because of the resistance which has been evolved. Finally, there are therapeutic strategies that are constantly being researched and advanced as the SARS-CoV-2 also evolves. So my slide has utilized quite a few different sources of information, which I have listed here. Some are very interesting reads and go into quite a lot of detail of how the mechanism of action of COVID-19 allows it to exploit its host, infiltrate the cells, and cause the overall infection. And I would really highly recommend reading through these if you were interested in learning about the details. So that's the end of this session, and I'm hoping that this has helped you understand variants, mutations, and how they evolve resistance. I have provided my contact. If you wanted to ask any questions, I'd be happy to answer those.